Okay. First, it was a pleasure to learn about the journey of Louise Harel. It was really nice. Thanks. And thanks for the organizing committee, Stefan and Claire, for the invitation to come here. It's really a great pleasure to be here and see so many of you. So the dark genome is not only long non-coding RNAs, consists also of regulatory DNA elements, and I'll try to discuss what we do with them. I'll mainly show you unpublished results after a short introduction. So regulatory DNA elements, these are just short sequences of typically not longer than 8 to 10 nucleotides in the genome that recruit transcription factors. And they, you can find them in promoters and enhancers. And by recruiting the transcription factors, they regulate gene expression of protein-coding genes, non-coding genes, and so on and so forth. Promoters, you know, of course, they are residing next to just upstream of transcription start sites. And enhancer, I'll just we'll show in a second what they are. It's important to know that transcription factors that are recruited to these regulatory DNA elements are key genes in cancer development. And I here listed a few of them that will show you some a little bit on those. That are transcription factors like p53, estrogen receptor, androgen receptor. Androgen receptor plays a role as a mitogen in breast cancer, androgen as prostate cancer, and YAP, TAZ, and TIAD that play a role in many cancers. They are amplified and correlate with aggressiveness of resistant cancer. Right, so enhancers, again, they are collections of regulatory DNA elements. They regulate gene expression from a distance, and they do it by DNA looping, and thereby they come close to the transcription start site. They regulate the transcription in orientation independent. They are marked by P300 and CBP binding proteins, which also modify histones. And in this type of modifications, and you can recognize those by just looking at those modifications. Furthermore, they express also enhancer RNAs, and we did some work on that, but I'm not going to discuss a lot of it, except that the amount of expression of enhancer RNAs correlates with the enhancer activity. So the more they are active, the better they express enhancer RNAs. Some of the enhancers, they may have function, but this is out of the scope of today. So we have, as we heard before, about 22,000 protein coding genes in our genome, and enhancers, we have maybe 500,000 or more. So the challenge is, of course, to identify functions of these enhancers and correlate them with certain phenotypes. We can measure their activity and correlate their expression. You can see here we can measure the activity of protein-coding genes on long non-coding RNAs by RNA sequencing. And we can measure the enhancer activity, as I mentioned before, by technologies that map or measure nascent RNA expression and enhancer RNA levels. So by this, you can measure this, and you can try to correlate the two and try to find relationships. However, which of the enhancers regulates which target and which is important, and that's the most important thing for the phenotype, that's really very difficult to annotate and map in a typical cases. So this is the more or less the structure of enhancers, promoters, protein-coding genes, and insulator or regions that are recruit CTCF and cohesin, and to form this type of DNA loops that brings enhancers in close vicinity to the protein-coding genes. And as I mentioned, we would like to set up genetic screens as are done for protein coding genes and long non-coding genes maybe in the future, and for those regulatory DNA elements. That's our goal. And the idea would be to, sorry about that, the idea would be to try to knock them out or knock them down and see what is their effect on a phenotype. So in a really 20 years ago, we developed this type of RNAi tools to knock down protein coding genes, but of course this is not suitable for this type of purpose. With the development of the CRISPR technology, we thought that they can be suitable not only for knockout of genes, but also to study those regulatory DNA elements or enhancers. And the idea is very simple. Take this CRISPR-Cas9, for example, technology, introduce it into cells and target really and that's key, only the elements, the motif elements, the consensus motif of the transcription factor. And by targeting this, we disrupt the binding of the transcription factor and locally affect the enhancer activity. So we've done this in the past 3 years quite extensively, studying many of the transcription factors that I just mentioned, including p53, estrogen receptor, androgen receptor, and CTCF. So just concluding those studies in short, I would say that at least for these cases, p53, estrogen receptor alpha, CTCF, we could use this CRISPR-Cas9 technology to functionally, genetically perform genetic screens. And we could conclude that they have the potential to uncover regulatory DNA elements and enhancers with tumor suppressive or oncogenic function. Identify their targets and thereby their function. Again, the key, I think the key success factor in these experiments was that even one nucleotide deletion or insertion to these elements disrupts almost completely the transcription factor binding site. And that's because the dimension of the motif are changing. So that was important. In the vast majority of cases, we could show at least 80 to almost 10-fold, 80% to 8-fold reduction in transcription binding capacity. Another interesting one, once we identified the enhancers that are functional in a certain phenotype, we could tile the entire enhancers and beyond with CRISPR, and by that identify other elements and other factors that might be important for that, for the phenotype. So we can mechanistically get insight into these regions. And maybe last but not least, another important thing is in this kind of screens is that we actually affect only the activation by the transcription factor. So only the extra effect on the target gene. We don't disrupt the essential function of the genes. And that can be very important in certain cases because if the gene is essential and knockout or knockdown of the gene affects cell viability, then it's not possible to identify this gene in any of the screens you may think of. However, if we just affect the activation, we can get the phenotype that we really want. I'll show you an example of that. So back to these key genes in cancer development. I want to focus today on this YAP-TAZ TID. YAP is a co-transcription factor together with TAZ, and TID is the DNA binding protein. And they belong to the Hippo pathway that was first identified in fly. And you may ask yourself, what is this Hippo fly? So I get from my postdoc, tells me this is the Hippo fly. No, it's probably not. It's only a fake one. This is the Hippo fly. And these are knockouts of kinases upstream of the this pathway, the EPO pathway, and this is a tumor suppressive pathway. What happens is that organs and cells start to proliferate and grow uncontrollable. And to jump to a more relevant picture of the EPO pathway in human cells, you can see that these kinases regulate other kinases. These are LATS1 and LATS2, and they phosphorylate these co-transcription factors, YAP and TAS. For the short, I'll call them YAP. Once they are phosphorylated, they are sequestered in the cytoplasm, and they are degraded, and therefore they are not functional. However, when this pathway is off, then a YAP is not phosphorylated, enters the nucleus, binds to this TID DNA binding, associates with the DNA, and activates transcription of many genes. The result of that is that the cells really change their phenotype, start to proliferate much more, they survive more different stress responses, they compete much better with the environment. The stem cell nest is more pronounced, they metastasize, and they can regenerate better. So really marked effects on cell phenotype. So we can find YAP activation in many aggressive cancer types. The option for therapeutics is limited because this is transcription factor. It's very hard to target, as we all know. And therefore, if we maybe find downstream targets that are important for the phenotype, we can try to target those. Not necessarily the enhancers, but the targets of those enhancers. So the group of Stefano Piccolo has performed quite some time ago, from the chip sequencing with this YAP and TID in different breast cancer cells, and identify the targets. And apparently the vast majority of the targets are active enhancers. So this TID with the YAP, when they are active, they enter the nucleus, they bind mostly enhancers and activate gene expression. So in my group, Lee, PhD student at the time, now has graduated and is staying for a couple of months more to finish this project. And Alejandro, they decided to look at this issue and try to identify downstream targets of YAP that are relevant for cancer in that perspective. So the way we design these CRISPR screens is the following. I didn't talk about it before that, but this is the way we do it. We first design an activity assay. This is the basic. Typically, we will use tissue culture cell lines, but also it can be done in vivo or other means. We then take a look at these chip sequencing data sets of the transcription factor, identify the binding sites and the motifs, and then construct a CRISPR library that targets these motifs only. And typically, we will have like 2 or more CRISPRs, up to 3, 4, 5 CRISPRs that can target this motif. This depends, of course, on the motif itself, as the CRISPR is restricted by the PAM, and GC-rich motifs are more easily targetable than the others. Okay, we then perform the screen, the functional screen, analyze the results, validate them, explore the underlying mechanisms, and exploit maybe, if possible, also for diagnosis and therapy. So setting up the biological system for YAP. We took MCF10A cells. So MCF10A cells are immortalized breast cells. They have a diploid genome, so we don't have amplifications and deletions, which is a better requirement for CRISPR screens, really. In contrast to doing it in cancer cell lines where you have these deletions and you can affect the CRISPR effectiveness. So here you can see that control cells, when they grow, they look epithelial, and they don't form any mammospheres, we call them, or these organoids, small organoids in 3D. However, when we introduce to them an active YAP, so this YAP is mutated in serine residues that normally are phosphorylated and render YAP inactive, we get this morphological transformation to more mesenchymal phenotype, and the cells grow as mammospheres. If we repeat it, I don't show you this result, but if we repeat the same thing with an inactive YAP, so containing the same mutations, but then one extra mutation that gets it inactive, in binding to TID, it's not functional anymore. And okay, so what is the effect? The basic of the screen that we are going to do is proliferation. So we ask, okay, what is the effect of this activated YAP on proliferation? If we grow the cells without any stress, so in normal conditions where MCF10A grow, we don't see any advantage. So this is a mix, a growth competition assay, when we mix two cells, two different colors, and we follow in this case, the MCF10A compared to the YAP cells. So we see that the MCF10A compete well, compete very well with the YAP. So they grow together, there's no advantage. However, when we remove EGF, or we do other different stress also, then we can see that the competitiveness of the MCF10A drops in comparison for the YAP. So now the YAP gets proliferation advantage. It continues to grow in the same rate, while the MCF10A, they stop growing more or less. Similar phenotype we see if we take this MCF10A, and this is important, and we knock out the activated YAP. So not the endogenous YAP, but the activated YAP. Again, we see that those ones are competing very poorly with the YAP cells. So this is important because this, in fact, is our positive control in the screen. You will see it later. We expect these guide RNAs against this activated YAP to be depleted in the assay with time. So this is actually, we call it like a drug addiction, or the cells became addicted to the YAP. They don't grow faster, but they are more sensitive. They are resistant to stress responses. So they ignore this type of stress, this damage that hyperproliferation or proliferation, unrestricted proliferation causes them. Right. So a little bit of details. So now we do the library. So we took this chipsec from the group of Piccolo, Stefano Piccolo, and identified all the peaks and the TID motifs that are there inside. This is the motif. This is where we will target the guide RNAs into it. So we had 2,900 peaks. 2,500 of them we could target with a CRISPR, and we created a library of about 5,000 guide RNAs, including positive and negative control. So we have like hundreds of negative control and some positive controls. This will be the YAP and guide RNAs against the active YAP. So to check the effectiveness of this guide RNAs, we picked up 3 top from the 10 top binding sites of YAP chipsec. We picked up the three randomly that had at least 2 guide RNAs targeting these enhancers. So these are the 3 enhancers. With the chipsec, you can see here, the chip qPCR, you can see that this is the binding to these enhancers. And this is a positive control. You can see that really they bind very well. So they are, because we selected them like that. But we selected them not according to phenotype, but according to the binding. These are negative controls. Then we introduced 2 guide RNAs every time for the same, the enhancer that they target. You could see that the binding of YAP to these regions is dramatically reduced. So this is a polyclonal population, and it's at least 2-fold to 4-fold reduction in each case, very specific reduction. So we conclude that this method works, and to interrupt YAP binding to these enhancers in a specific way. So here's the design of the screen. take these MCF10A cells, introduce them to the library. We start to grow them, grow them as a dish in time for 20 days, collect the genomic DNA, we PCR amplify those guide RNAs, and we sequence them, and we look for the ones that drop out, specifically in the MCF10A cells, YAP, compared to the MCF10A cells. All right, so this is more or less the results. If we compare the two cell populations, in blue you can see the controls. I think in orange, red, these are the positive controls. So all these YAP, active YAP, guide RNAs against active YAP. You can see that they all shifted to the left in this MCF10A YAP, but remain the same, almost the same in this MCF10A. So really worked. And then in green, these are guide RNAs that show this shift, statistically significant shift in MCF10A YAP cells, but not in MCF10A. So from this list, we picked up the ones that had at least 2 guide RNAs that targeted those enhancers, and we continue with those. From these 10, we looked also at growth-seq. I will not bother with this, but we selected 7 that showed in other systems activation of these enhancers in endogenous YAP conditions in aggressive breast cancer. So we selected those 7. We first cloned their enhancers. We just simply subcloned them into a luciferase vector downstream of the reporter. So this is placed as an enhancer in this plasmid, and we check the activity compared to control, and we clone them in 2 different orientations, forward and reverse. And you can see for all these 7 enhancers, they could upregulate, activate enhancers. So they activate the reporter system, indicating that they are enhancers. What is their effect on cell proliferation? Now we created polyclonal populations of single guide RNAs, and we examined in first instance their mutations rate. So you can see here in these polyclonal populations, we have around from 95% to about maybe the lowest 65% mutation rate in those enhancer regions. So it's very effective. And the result of the cell proliferation is reduced proliferation of these MCF10A cells, not as much as the positive control here, which targets, again, the active YAP. So these are lower but significant, and no effect completely on MCF10A. So this is very specific to the YAP function. Right, so what are the target genes of these enhancers? So this is a bit of a global view, a bit dense, but here you can see the names of the target genes. See here MYC and cyclin D1. These are very well-known targets of YAP. So there we fished out the enhancers, which were not known. The enhancers that potentially activate those genes. And here you can see in more small, I'll zoom in a second, RNA sequencing results, where you can see that when we compare these cell populations to control cells, and you can see in green the genes that went down by the guide RNAs, both of them. So both guide RNAs. So you can see here those genes. So this way we could identify the potential target genes that are affected by those enhancers, and they are listed here. What is apparent here is the red one is the TAD, the chromatin structure in these regions, and you can see that in this case, for example, the enhancer here in yellow is in the middle of the gene, and this is the TAD, and it's in the intron and control this gene TREM2, and we can see it with all of the others. Sometimes it resides here, sometimes it's right downstream, sometimes in the intron, but always inside the TAD. And we don't see significant effects on other genes in the vicinity, in gray. So from this for 7 we could identify enhancers, 6 we could identify target gene. We correlated with TCGA data sets to see which one of them correlates with the transcription, with the RNA levels of YAP in different tumors. We can see that there is no correlation with GAPDH expression, as expected. There's some correlation with cyclin D1 and MYC expression. We think that these are regulated by YAP, but also they are regulated, of course, by E2F and other factors. These important genes. This is why they are correlated, but not so much as others, as you can see with YAP. However, this SLC39A1 and mostly this gene TRAM2 correlate almost invariably with all, very significantly in all tumor types. And we think those are actually maybe solely regulated by YAP. And we focused on TRAM2 here, just because it was the top correlated gene with YAP. Yes. So this is again the cheap qPCR of the TRAM2 locus, enhancer locus, and you can see here again it binds very well YAP, but when we knock out with these two guide RNAs the enhancer, again this drops really much. The binding of YAP drops really much. So they are very effective, those two guide RNAs against the enhancer. We also measure the enhancer RNA and the activity of the enhancer drop down very much. The effect on the protein is also the same. When we knock out TRAM2 enhancers, we can see a drop of TRAM2 levels, and we can also overexpress it or express it in these cells ectopically, and we can rescue this effect. Now the effect on the proliferation is, again, as before, we could see the knockdown or the knockout of this CRISPR of this enhancer, TRAM2 enhancer, affects proliferation, and when we re-express TRAM2 back, we can rescue this, indicating that TRAM2 is possibly the sole target of this enhancer that is related to YAP activity and this proliferation assay. Right. Also, we can show that in fact there is a loop. This is the enhancer. It sits in the intron of TRAM2. This is the promoter, and using different combinations of primers in a 3C chromatin conformation assay, we can show that it's here all that the promoter really interacts with the enhancer and not with downstream or upstream regions. So this is a very specific interaction of the enhancer with the promoter, as we expect, which confirms the effect of this enhancer and the promoter on the TRAM2 expression. Right, and this is also, I think, a nice and interesting point, is that if we now knock out TRAM2 itself, we can see very dramatic effects on the proliferation of YAP, MCF10A YAP cells, as expected, but also they affect the MCF10A. And that's because TRAM2 has probably essential functions in these cells. You can see the effect on protein expression. Right, so we wanted to know what the effect of TRAM2 on the protein expression globally. Thinking that there will be a very nice correlation, we hoped for a very nice correlation with proliferation markers. However, we did see some effects on mitotic proliferation. These are the YAP cells and TRAM2 overexpression cells. Now we overexpress the TRAM2. But the most pronounced really group of genes that changed was the epithelial mesenchymal transition. YAP, we knew that it really creates this, I showed you that, this creates this phenotype, mesenchymal and aggressiveness. TRAM2, we didn't know, and it really creates this phenotype. Even if we now take these YAP cells and we knock out, again, the TRAM2, which I showed you before, we can see that this group of genes now going down very, very dramatically. And the cells lose their well, I'll show you in a second. Seems that the most pronounced gene group is the mesenchymal, epithelial to mesenchymal transition. One of the genes was vimentin, a well-known gene in the yes. You can answer. No? A well-known genes in the mesenchymal. Oh, it's a timer. Okay. Good. Mesenchymal, I'm almost done. And also when we knock out TREM2, we can see that YAP. I'm only 30 minutes. Okay. Okay, knockout, we can see that this TREM2, the vimentin goes down in these TREM2 knockout cells. So this will suggest that TREM2, like YAP, has influences on mesenchymal and the way that cells migrate and invade. This is only to show you in brief that it's really the case. In vitro migration, we can see stimulation of the cell's movement with microscopy by the YAP cells, but also by the TREM2 overexpression. We also see that in vitro invasion is dramatically increased by TREM2 as much as YAP. And also in vivo, when we inject these cells and look with the window into the cells, we can measure their activity, their migratory and invasiveness. We can see these TREM2 cells really migrate and invade as much as YAP cells. So they really show this phenotype. Now, if they do that, and it's important for metastasis, then they should show also correlation with survival. And this is, again, this TCGA data sets, pan cancer, all cancer. And it was known that YAP expression correlates with poor survival, but also TREM2 correlates exactly the same with poor survival. It's almost identical, but you can spot the differences. So really, TREM2 is kind of marker of YAP and metastasis and poor survival. Now, okay, I skipped what TREM2 is, but TREM2 is a translocating chain-associated membrane protein. It basically sits in the ER membrane and translocates proteins from the cytosol while they are translated. So it's cross-translational to the ER itself and switches actually from the ER membrane proteins from the cytosol to the luminal of the ER. This also creates their expression. It stimulates their expression and stabilization, and it depends on the real client depends on signal transduction peptides. So the first thought we said, okay, maybe this vimentin is one of the clients, but it's not. It doesn't have any signal transduction peptide. So we did perform this again, this proteomics analysis of TREM2 overexpression, knockout, what I showed before, and YAP, and ask what is now common to these proteins, what is common group for all these conditions, and these are the genes, the proteins, and in vast majority of them have a signal transduction. These are the green ones, and even the best one is this FSTL1 is really connected to vimentin, to metastasis, and to migration. So we think that this is really the key target of YAP via TREM2. Right, so just in two slides more. Of course, you can inhibit this translocon, not directly TREM2, but associated processes. And we ask, okay, YAP is now involved in this aggressiveness and resistance to drug treatments, whether messing with this TREM2 function will make these cells more sensitive. So these are preliminary results. We have to do more experiments, but I'll show you just one thing that Sebastian has did. We took these melanoma cells, and if you treat them with memorafenib, they, of course, are very sensitive. Also, the TREM2 is very sensitive, but if you express YAP, they are highly resistant. And then if we now take and we block now the translocon, we can see that these TREM2 cells, they are highly sensitive, and now the YAP cells, instead of being resistant, they become sensitive again. So we think the combination is or alternating treatment of inhibiting the signaling and then inhibiting the translocon may affect this melanoma. We have, of course, to do more on this, and we are doing. these days, but we think this way. So let me just wrap up this story. Is we did enhancer screens to identify targets of YAP, and we found many that are in proliferation, and also some that connects migration and proliferation together with this scheme. And maybe more general one is that indeed multiple enhancers are mediate really YAP-induced proliferation. And TREM2 regulation by YAP is required for proliferation, and also EMT and migration and invasion. And maybe synthetic vulnerability, we uncovered this synthetic vulnerability for YAP activation in cancer. Right, so I mentioned the people that have done this work, and also our collaborators here, facilities, and our sponsors. And thank you for listening. And if there is any question, I would be happy to answer. Thank you very much.